In this chapter, we're going to get into proteins, starting with their building blocks, amino acids, and talking about how they build up from there. So, amino acids, as the name suggests, have an amine group and a carboxylic acid group. Now, we're specifically looking at alpha amino acids. Those are kind of the key ones that we use for building proteins. And they're so called because both the amine and the carboxylic acid are attached to the same carbon. In other words, they're both at the alpha position. All right, so that carbon also has a hydrogen and another group attached to it. So note that we have four different groups attached to that carbon, and so that is a chiral carbon. Now, the amine of one amino acid and the acid of another amino acid can link together to form what we call a peptide bond, which is essentially just an amide. All right, we basically link the nitrogen from one amino acid to the carbonyl carbon of another. Okay, and so we form an amide group there. All right, so if you link together, short chains of these, we call these peptides. And we can add prefixes before the word peptide to state how many amino acids we have linked together. So, for example, a dipeptide has two amino acids, a tripeptide has three, and so on. Okay. Once we get to even longer chains, we start building these peptides into proteins. We start getting into proteins that have specific structures that tie into their function. And we'll talk more about that as we go on in this chapter. Okay, so the key difference between our amino acids is that side group. So all amino acids have that amino group and they all have that acid group, as I pointed out. And so that carbon has a hydrogen and some other group. So we just generically call that R, but basically it doesn't have to be an alkyl group. Okay. Now we can break this down into different subcategories to make this a little bit easier to group together. So you could break this down into like polar versus nonpolar groups. So nonpolar groups are mostly carbon and hydrogen based. All right, as the name suggests. The polar groups can be subdivided even further. We can describe them as groups being acidic if they have a carboxylic acid group, like aspartic acid over here. They could be basic if they have an amine, okay? And if they are neither, acids or amines, but do have some other polar groups or, are capable of hydrogen bonding, as we can see here, they're called that. Now, I mentioned earlier that that carbon that connects all of these groups together is chiral because we've got four different groups on it. Um, Basically, the L enantiomer is the prevalent one you'll find in nature. So I think it's safe to say that unless you're told otherwise, you can assume that the stereochemistry around that carbon is going to be, it would give you the L enantiomer. Okay, so you can assume that as you can see here, if we list our carbons in order, going with our acid group at the top, and then our side chain here, our R group at the bottom, the amine and the hydrogen are, your two groups attached to that chiral carbon in your Fisher projection. So having the amine would show up on the left, basically, to give you the L enantiomer. Out of the 20 amino acids that I mentioned are available, they come from a variety of different sources. Now, the body can make about half of those. So in order to build up the remaining, your proteins out of the remaining 10 amino acids, you'd have to get them from other sources, basically through diet. Okay, so essentially through protein-rich foods. Now, while most amino acids can be found in proteins, and that's why you can get them through digesting proteins in protein-rich foods, not all amino acids come from proteins. Two examples here. You can see that we've got GABA, which is a neurotransmitter. GABA actually isn't an alpha amino acid. Notice that the amine group and the carboxylic acid group are not attached to the same carbon. Okay, here's thyroxine, which is a hormone found in the thyroid, and that is an alpha amino acid. Notice that the both the carboxylic acid group and the amine group are attached to that same carbon. Okay, but of course you won't find it in a protein. It's not an amino acid you'll find linked up to other amino acids to make a peptide. All right, so now your carboxylic acid group and your amine group are both capable of proton transfers. They're capable of behaving differently in different protonated or ionic states depending on the surrounding pH. Okay, so basically, if you're under acidic conditions, your amino acid, you can think of there being an excess of protons in solution, so your amino acid will be completely protonated. So what that means then is that while your carboxylic acid group is neutral, all right, it's got its proton attached to that carboxylic acid group, your amine group actually picks up an extra proton and becomes an ammonium ion. All right, so notice that overall we wind up having a positive charge. Now, under basic conditions, where you have a shortage of protons, the reverse happens. Your ammonium ion is down to the neutral amine, right? You don't have that extra proton. But in addition to that, our carboxylic acid group is also deprotonated to give us a carboxylate ion. And so now notice that our amino acid is has an overall negative charge. Okay, now if you're in between these two, and this typically occurs at physiological pH, usually you're in between the two pKa's, that require either being fully protonated or fully deprotonated. But if you're in between those two, we have what's called a Zwitter ion. Okay, so our ammonium ion is protonated, and our carboxylic acid is deprotonated as a carboxylate. And so notice that we have a positive and a negative charge. All right, so the net charge is neutral, but we do have two charged parts to our molecule. Okay, and that's what a Zwitter ion is. So here's a list of a few common amino acids, and you can see the pKa's for the carboxylic acid groups and the ammonium groups that you'd find on these amino acids. Things get a little bit interesting when you have either an acidic or a basic side chain. So you can see here where you've got a non-polar or a hydrogen bonding side chain. We don't have one listed, but you have to also consider the pKa of those side chains. Okay, so the thing with a Zwitter ion is that it's amphoteric. It can act as either an acid or a base because you have two parts of that molecule that are respectively acidic or basic, right? Your ammonium ion can act as an acid by donating that proton if there's a base present. Okay, now if you have an acid present, the carboxylate group can act as a base, picking up that proton, giving you the neutral carboxylic acid. Okay, notice over here when your ammonium ion acts as an acid, notice you get the neutral amine as well. Okay, so let's practice this. Here we have we have glutamic acid, right? So that's an amino acid. Here's our amine group. Here's our our carboxylic acid group. Notice that it has an acidic side chain. So notice that we have a carboxylic acid group over here. All right, so if we look at glutamic acid, note that the side chain has a pKa of 4.25. So what is this molecule going to look like at a pH of 3.5? All right, so 3.5 is a pH that's lower than your ammonium ion being protonated, which makes sense, and it's above the the pKa of your carboxylic acid group. Again, that makes sense. So that tells you that basically this group is going to be deprotonated and this group is going to be protonated, right? But what about our side chain? Notice that our side chain has a pKa that's above the pH. So our pH is relatively acidic compared to the pKa of our side chain, which means that this this carboxylic acid group here is not going to be deprotonated. It's going to be protonated. That proton is going to stay on our carboxylic acid group. Okay, so unlike the main carboxylic acid group that's attached to that alpha carbon, while that one's going to be deprotonated, the one that's attached over here will remain protonated. So in a nutshell, it'll look like this. Basically, we will keep this protonated. This is actually going to be a cation, so we'll have a positive charge here and 3 hydrogens here, and then this proton's going to be missing, and there's going to be a negative formal charge here. Okay, so but this one stays neutral. So we still have a Zwitterion here, but that's kind of what that looks like. And again, please note how that agrees with our pH of 3.5, right? So if your pH is below the pKa of a group, that means that that group's going to be protonated. If it's above, if your pH is above the pKa, that means that group is going to be deprotonated. Okay, and so that's what we observe here. This group is deprotonated. These other two groups are protonated. All right, so when it comes to describing your amino acids, especially when it comes to discussing is something going to be protonated or not? The term you'll often hear brought up is what's called the isoelectric point, or PI. All right, now the isoelectric point is the pH at which you kind of have a perfect Zwitterion. You're really in between, your pKa's of both your acid and basic group. All right, and that's actually how that value is calculated. You literally just take the mean of those two numbers. All right, so that's pretty straightforward when you have a neutral side group. Okay, now things get a little bit trickier if you have an acidic or basic side group. What you've got to do is you have to pick the two groups that are similar. So if your side group is basic, you use the pKa of your amine or ammonium group on your carboxylic acid, on your amino acid, and you take the average of those two pKa's. Likewise here, if your side group is acidic, you take the pKa's of your two carboxylic acids and take the mean of those two numbers. All right, and so again, remember this in all three cases, your isoelectric point basically gives you the pH at which you kind of have the largest concentration of your Zwitterion. So it's kind of like the pH at which you make the perfect Zwitterion, like sort of dead, in the middle between, being sort of neutral effectively. All right. So with that in mind, armed with these PI values, and knowing that you've got a system at a particular pH, you can figure out whether your amino acid is going to be effectively neutral as a Zwitterion, or if it's going to have an overall positive charge and be cationic, or have an overall negative charge and be anionic. Okay, this gets really useful when you want to separate out a mixture, or just to help identify the components of a mixture of amino acids, because if you put them on a gel and place it under an electrical current, ions will migrate to the oppositely charged electrode. So your anions will migrate to the your negatively charged anions will migrate to your positively charged anode. Your positively charged cations will migrate towards your negatively charged cathode, right? And then, of course, your Zwitterion, if you're at the isoelectric point for a particular amino acid, it's going to kind of stay in the middle. All right, so you can see this example here with 3 amino acids of different isoelectric points. We're doing this at a pH of 6, and alanine's basically at its isoelectric point. So it kind of stays in the middle here. Whereas glutamic acid has an isoelectric point of 3.22, this pH is above that isoelectric point, which means that, relatively speaking, this pH seems basic compared to this PI, right? So effectively, you're going to deprotonate that glutamic acid as much as possible. And if you're deprotonating it, that means it's going to be relatively negatively charged. It's going to be an anion, and that's why it gets attracted to the positive electrode. Conversely, here we have lysine with a pI of 9.74, so the pH is lower than that, which means that the environment is relatively acidic. So this is going to be extra protonated. It's going to have extra H plus ions on it, which is going to make it overall positively charged, or cationic, and therefore it gets attracted to the negative electrode, and that's why it migrates to that side. All right, so that technique's called electrophoresis. So basically you put your medium in a gel and apply electrical current at a particular pH, and that's how you can separate out your your mixture of amino acids. And so you can kind of see where the bands of those amino acids show up, and based on the pH you can figure out the pI, the isoelectric point of that amino acid, which in turn helps identify them. Now, of course, this is just to identify the amino acids. If you really want to, on a larger scale, separate out a mixture of amino acids, you can use techniques we've already seen before, such as column chromatography. All right. Now, if you want to visually identify the presence of an amino acid, we can use what's called an anhydrin. Anhydrin can be used as an indicator. Basically, when it comes into contact with an amino acid, it produces a brightly colored product. This product has a purple color that's readily visible, is easily identifiable. And so basically, if you spray it onto a location and that location turns purple, you know that there are amino acids present. Okay, so this is often used, for example, when you're effectively sort of dusting for fingerprints, so to speak. It's the traditional method involved an actual dust, but you can look for you can essentially spray for fingerprints by using an anhydrin spray. Okay, so let's look at some reactions that involve amino acids. So let's start off by looking at how to make amino acids. Now, in an earlier chapter, I mentioned the awesomely named Hell-Volhard-Zielinski reaction. And so let's talk about that again, right? So basically, we start off with a carboxylic acid. All right, we can add a bromine at that alpha position using bromine, molecular bromine and phosphorus tribromide. And then using, ammonia, we can basically do a nucleophilic substitution and swap out that bromine for an amine. So again, notice that the, ammonia is relatively nucleophilic. You've got that lone pair on that nitrogen, and bromine's a great leaving group, right? So that's a relatively straightforward substitution reaction. Another way to make amino acids is to use the amido-malonate synthesis, which is very similar to the malonic ester synthesis that we saw back in chapter 22. Essentially, you start off with diethyl amido-malonate. Okay, so it's kind of similar to our malonic ester, except instead of some R group over here, we have our an amide attached to it. Okay, so you react that with a base. Again, like with the malonic ester synthesis, you want to pick a base that's similar to your alkoxy groups on your ester, and then have that react with a with an alkyl halide. So again, your base deprotonates that alpha carbon really easily. And that makes it a nucleophile, so it just does a nucleophilic substitution with our alkyl halide and just basically tacks on that alkyl group. Then you can just heat that up with dilute acid, and you basically hydrolyze this into the resulting amino acid. Okay. Another method is the Strecker synthesis. So basically, you start off with a an aldehyde reacted with ammonia, basically, and you can get that ammonia by taking ammonium chloride and just having ammonium chloride present that can sort of decompose into HCl and ammonia right there. But basically, your ammonia acts as a nucleophile on your carbonyl carbon, and so that intermediate gets, picks up a proton. Basically, you have two proton transfers, right? So you protonate that carbonyl oxygen, it becomes an alcohol group, and you deprotonate that ammonium ion to make your neutral intermediate here, right? So at that point, then you can protonate your alcohol group, make it a good leaving group. Okay, you can see here that we lose water essentially and form the corresponding imine. You can then tack on if you have a cyanide ion, so like in the form of potassium cyanide or sodium cyanide, that's your nucleophile here, and we basically get this nitrile group, which we can then hydrolyze into the corresponding carboxylic acid group. Okay. Now, you probably noticed with those previous, with some of those synthetic methods, we're going to get a racemic mixture of our amino acids. You typically go through, where that carbon's sp2 hybridized, and so you're going to get your nucleophile coming in and adding on to either side of that carbonyl carbon, and so you're going to get a racemic mixture. Now, this is problematic because, as I pointed out earlier, that alpha carbon is chiral, and in nature we tend to get the L enantiomer pretty much exclusively, right? So if you want to use this for biochemical applications, you probably want that L enantiomer, and a racemic mixture isn't going to be helpful there. Okay, so we need to find out how to either, basically separate out our racemic mixture into its two enantiomers and then save the one that we want, which can be kind of difficult, right? You need basically you need to do run a column chromatography with a sort of chiral resolving agent. Okay, and that kind of basically helps select out one enantiomer over the other. What might be preferential is to not even get to that point. Don't even make the racemic mixture to begin with. Be selective in making just one enantiomer, what we call an asymmetric synthesis. Okay, so that's generally preferable because it eliminates the problem before you even get to it. So to do that, though, you either have to start off with an enantiomerically pure reagent, which can also be problematic because if you're going through a sp2 intermediate, you're going to lose that chirality. So actually that method doesn't work in that situation. So what might be better is to use a catalyst, use a chiral catalyst that has a lower activation energy for the product that you want. Okay, so to do that, one way to do that is to use Knoll's catalyst. All right, so if we remember back in chapter 9, we kind of talked a little bit about asymmetric catalysis, and Knoll's catalyst is one that does that. All right, so basically you can take this alkene over here and add hydrogen across it, but specifically add it to one side. And that's what causes this configuration alone to be formed. So you get the S enantiomer, not the R enantiomer. Okay, and of course, then you can take that product and go on to convert it into the corresponding amino acid. All right, Noyori's catalyst is another good example here. So I think I talked back in that chapter about BINAP. If you recall, the two hydrogens that are sticking out here on these two rings can't exist over here in this space. That's just too narrow for both of them. And so due to sterics, these two rings can't be flat. One of them gets pushed towards you, and one of them gets pushed away from you. So you have like kind of a corkscrewing appearance to that to this ligand, to this group that's attached to the ruthenium. Anyway, my point here is that this that's what makes this chiral, right? And so it selectively, again, puts the hydrogen on only one side of this carbon-carbon double bond. Okay, and so we see here that we have a 99% enantiomeric excess, right? So, we have like 99, you know, 0.5% of our product is this enantiomer, and only 0.5% is the other enantiomer, right? Yeah, and again, you can take this product and do a few more reactions, and you convert this into the corresponding amino acid. Okay, so now that we've talked about making amino acids, let's talk about linking them together to form peptides. All right, now I mentioned a tripeptide means there are three amino acids linked together. Okay, so here we have glutathione as an example of tripeptide. It's made up of glutamic acid, cysteine, and glycine, all linked together. Okay, and again, notice we have these peptide bonds, which essentially are amide bonds. We have the carbonyl from the carboxylic acid of one of our groups connected to the to the nitrogen of the amino group of another amino acid, right? And that's where our amide bond is. Okay, and again, here's another amide bond. Here, the carbonyl, from the carboxylic acid of this amino acid is connected to the nitrogen of the amino group of this amino acid. Okay, now notice that both ends, the amino group and the acid group, are both involved in connecting two groups. But since every amino acid has both of these groups, the order is kind of important because you have two options now for linking together two amino acids. So to give you an example, let's say we're trying to link together alanine and glycine. We could have the alanine, the carboxylic acid, or the carbon end of our alanine connected to the nitrogen end of our glycine. Okay, or we could have the carbon end of our glycine connected to the nitrogen end of our alanine. Okay, so going from left to right, from our nitrogen end, our amino end, to our carboxylic acid end, or carbon end, we could have alanyl glycine, okay, or abbreviated as ALA-gly, okay, or vice versa. We could have glycyl alanine, right? So where we have our glycine on the left and the alanine on the right. So the nitrogen end is also called the N-terminus, and likewise the carboxylic acid and the carbon-based end is called the C-terminus. And by convention, we typically write the N-terminus on the left and go from there to the C-terminus. All right. Now, if you have trouble remembering that, just remember that we're dealing with amino acids, and so the amino end is on the left, the open amino end is on the left, and the open acid end is on the right. Okay, as I pointed out with the example of glycyl alanine, we can use three-letter abbreviations for our individual amino acids. So glycine's abbreviation is GLY, alanine's is ALA, and so we could just write that like that. Now, regarding the amide bond itself, the peptide bond that's basically an amide bond, you need to remember that that nitrogen does have some double bond character to it, or it is sp2 hybridized because that carbon-nitrogen bond has some double bond character to it, as you can see from this resonance structure. Okay, so it's kind of easy to forget because usually you just have it drawn as a single bond, and so you would think there would be free rotation around that single bond, but there's actually a larger energy barrier to that than you would expect, somewhere in the ballpark of about 80 kilojoules per mole. So just realize that you can get that your peptide bond locked into one conformation versus another. Now, the S-trans conformation makes more sense from a steric standpoint. So that's usually the one that you often see. All right, so keep that in mind in your polypeptide chain that at least these the nitrogen carbonyl bond is kind of locked in that conformation to that S-trans conformation for the most part. That being said, you can also you can rotate around the carbon-carbon bond, so there's some free rotation in there. Okay, other things that can influence the rigidity of your polypeptide chain include the formation of disulfide bridges. But this can have a larger effect. We're going to talk more about the overall structure of proteins in general, and these disulfide bridges do come into play there as well. But for now, let's basically just point out that if you have a thionyl, side group, you could basically form a bond between the two sulfurs of those thionyl groups, and that's where we get our disulfide bridge. And now these two amino acids are locked into place here. Okay, so here you can see we've got a disulfide bridge within our peptide chain. So this is intramolecularly that we have that connection. All right, so it's an intrastrand disulfide bridge. Here we have two different polypeptide strands, and our disulfide bridge connects the two of those. So that is an interstrand disulfide bridge. So we've seen that we can, basically the order in which we sequence our peptides, the order in which they are linked together is kind of important. Even with just two peptides, with two amino acids, just flipping their order makes a different molecule. And we see that in larger proteins as well. So here's some examples of some peptides you'll see in biology, and you can see that just changing one amino acid in the chain gives you a different protein. Okay, now you can get some molecules that have similar functions, but if we look at this example here with vasopressin and oxytocin, you can see that just changing two amino acids in our chain gives us two different proteins with two different, completely different functions. All right, so again, we'll talk more about the different levels of structures to protein, and this is like the most fundamental. This is the sequence, the order in which our amino acids link together. But I just want to kind of stress that that's kind of our starting point for understanding how structure affects proteins. Okay, so given that this sequence is pretty important, let's talk about how we can accomplish it. How do we figure out the order of these amino acids in our peptide chain? So to do that, one method we can use is called the Edman degradation. Basically, we clip off an amino acid one at a time, and in a particular order. We start at the N terminus and work our way towards the C terminus, and in doing so, we basically can analyze each of these peptides each of these amino acids we clip off, and therefore we can get the sequence. So we start off with the amino group on our amino acid acting as our nucleophile and attacking the carbon on our phenyl isothiocyanate. All right, so it attacks that carbon, basically forms this intermediate, and then we have an intramolecular nucleophilic attack. So that sulfur becomes a nucleophile and attacks the carbonyl of our peptide bond. Okay, so when it does that, it basically closes this ring. We get this ringed intermediate. We do a proton transfer here, and when basically that oxygen can come down and reform a carbonyl, and then so kick off this group from the rest of our peptide chain. Okay, so notice that this nitrogen of this amide bond becomes the new N terminus. Okay, so we have this group, basically just leaving the rest of the peptide chain. So you can see again we have a new N terminus. Now this leaving group can then be, essentially analyzed, because this R group is kind of what we're looking for, right? We're trying to use that to identify what our basically what our side group is, and that'll help identify what that amino acid was. since you're dealing with really long peptide chains, oftentimes it's more convenient to use to sort of automate this process, and there are machines that do that, known as peptide sequencers. All right, so once you start getting to more than 50 amino acids, of course, this process gets, well, very just not practical, right? So what we need to do is we have to take our peptide chain and break it down to smaller, more workable chunks. Okay, so now, that being said, you can't just break a peptide chain randomly, because then you don't know how the different pieces go together and you lose information that way. So we need to select, we need to select a method of breaking up our peptide chain selectively. Basically, break it down in methods that we expect, and then that way we can, when we get the data at the end, we can take those pieces and put it together like a jigsaw puzzle. So we use what are called peptidases. These are enzymes that break down our peptide chain in specific spots. All right. So, for example, trypsin is a peptidase which will hydrolyze your peptide bond, your amide bond, specifically to the right on the carbonyl side of lysine or arginine. All right, so here in our sequence, here we've got a lysine, here we've got an arginine, and it's on the carbon side of that lysine amino acid that we cleave here. And likewise, here on the carbon side of that arginine, we're going to cleave here, and therefore we get these three shorter strands that are then easier to sequence using the Edmond process. Okay. Now later on, when we're trying to put this together, we'll know that out of these three strands, we know that this one over here must be on the C terminus side, because there's no lysine or arginine on the end here. Okay. Another example of a commonly used peptidase is chymotrypsin. That will cleave either on, by phenylalanine, tyrosine, or tryptophan. Okay. Now, let's say we're trying to do this backwards. Let's say we want to make a peptide. We want to make a sequence of peptides. So we basically take our amino acids and we want to link them together. Now, if you recall, the order in which we do this is very, very important. So we need to figure out how to not only link together these two amino acids, but how to link them in the order that we want. Okay, so first let's talk about the linking part. To do this, we use what's called DCC, or dicyclohexyl carboimide, right? So we have these, a double imide functional group here in the middle, and these two large bulky alkyl groups. All right, so what happens is basically once you deprotonate your carboxylic acid, your carboxylate, basically attacks the carbon of your DCC, all right, and you kind of form this intermediate where the, your carbonyl is prone to an attack from your amino group of your other amino acid. Okay, and when that happens, you now have a really good leaving group here, right? Basically, that leaving group plucks off the oxygen of your original carboxylate group, and notice that now when you reform this carbonyl, you've essentially formed that amide bond, or that peptide bond that we were looking for. Okay, so notice that we formed that carbonyl carbon, that bond between that carbonyl carbon and the nitrogen, and we've taken away the oxygen of that carboxylate group. Okay, now of course this links together any amino group and any acid group, but it just like with our mixed aldol reactions, there's lots of options here, right? If you have two if you have two different amino acids, you could have amino acid 1 combining with 2, you could have 2 combining with 1, you could have 2 combining with 2, or 1 combining with 1. So there's 4 different possibilities here, since the sequence is important. Changing the sequence gives you a different product. All right, so obviously we want to control this, because our yield of our desired dipeptide here is going to be awful. We're talking about 25% at best, if we don't try to control this. So what we want to do is we essentially want to use protecting groups. We want to block the amino group of our N-terminus, and we want to block the carboxylate group of our C-terminus and prevent them from reacting, right? We want them to remain termini in our final product. Okay, so we protect those groups respectively, we protect those ends respectively, and then we can couple together the remaining carboxylic acid group and remaining amino group using DCC. So what do we need for protecting groups, right? So how do we swap out our, or protect our amino group and our carboxylic acid groups? And you're going to find out that it's not the same protecting group. You need to have a different approach for each. So to protect an amino group, you can do so by converting it to a carbamate. Carbamate is much less reactive than the bare amine. Now, to do that, we react our amine with a BOC2O reagent. So what do I mean by that? So here we have ditert-butyl dicarbonate, where the this tert-butoxycarbonyl group over here that makes up half the molecule, that's abbreviated as BOC or BOC. So you'll often see this represented as BOC2O, okay, or BOC. It usually BOC's written in parentheses like this, but basically this oxygen here is this middle oxygen here, and this is our BOC group. Okay, so it's a tert-butoxycarbonyl. All right. So what we have going on here is that our amine attacks the carbonyl of this BOC group. All right. And when this carbonyl reforms, it kicks off the other BOC group, taking the oxygen with it. Okay. So when that leaves, here's that BOC group floating around, and it's resonance stabilized, of course. Now that can also, act as a base, right? We can deprotonate that nitrogen and make a more neutral intermediate. Okay, so here is our resulting neutral carbamate, right? So we have effectively, it's kind of like a combination amide and carboxylate, like an amide and an ester, effectively. So you've got a carbonyl carbon, and there's an oxygen and a nitrogen connected to it. Okay, so this BOC group is, it's a good protecting group. This prevents this nitrogen from being nucleophilic and stops it from reacting. All right. Now, the other reason that these BOC protecting groups are very useful is that when we're done with our sequencing, when we're done synthesizing our peptide, we need to return that amino, that N-terminus back to being an amine, right? So it's actually relatively easy to remove that BOC group once you want to. To do that, you use trifluoroacetic acid. And basically that will protonate the carbonyl oxygen of your BOC group. Okay, and essentially you'll have your tert-butyl group on your BOC group leave, and eventually that will get converted into a 2-methylpropane. The carbonyl, the carboxylate group on your BOC group that's attached to your nitrogen will eventually leave as a carbon dioxide, right? And this, of course, is very one-directional, right? That leaves as a gas, so you're not going to do the reverse reaction. And ultimately, what you're left with then is, of course, that amine group. All right. Another method to protect the amine is to use an FMOC group. That stands for 9 fluorenyl methyl chloroformate. Say that 10 times fast. So anyway, we've got this bulky protecting group. Again, kind of works very similar to your Bach group, right? You're making a a carbamate here, and that means that nitrogen is less likely to react. Okay. So to remove your FMOC protecting group, to do that, you typically do this with a base. So something like piperidine will work. They'll essentially, just pluck off that proton here, which gets this going, and the resulting carbanion. You kind of form a double bond here, which cleaves this carbon-oxygen bond, which reforms this carboxylate, which of course then can leave as carbon dioxide. All right, so there's all the products that you get out of that process. So here's that FMOC group with that double bond formed here. You can see here that when this bond forms, we also break this carbon-nitrogen bond, and there's the resulting amino acid. Okay, so to protect the carboxylic acid group, we can simply convert that into an ester. If you recall from our chapter on acid derivatives, esters are less reactive than carboxylic acids, and so converting our carboxylic acid into an ester will generally help that there. Okay, so one way to do that, of course, is to just use a the alcohol that we want for our alkoxy group that we're going to put on here, react that in the presence of an acid, and generally we can make an ester. The most common kinds of esters involve using either methanol or benzyl alcohol to make the corresponding methyl or benzyl esters. All right. Now, when you want to deprotect that ester and go back to the carboxylic acid of your amino acid, you can hydrolyze it using either a base or a dilute acid. So, normally you would use a concentrated acid catalyst to do the forward reaction to make the corresponding ester. To do the reverse reaction, you'd want to use a dilute acid so that you're going to replace this, that alkoxy group with an OH group. Okay. In the case of the benzyl ester, in addition to using, an acid, dilute acid, we could use hydrogen over a platinum catalyst. That will also, return this back to a carboxylic acid. Okay. In terms of an acid combination, for the hydrolysis here, another really good one would be hydrobromic acid along with acetic acid. Okay, so we have to add these extra steps in. So 2 extra steps for each amino acid, a protecting step and a deprotecting step. And that's just necessary in order to selectively produce the amino acid or the peptide combination that we want. Okay, so even though we're adding 2 extra steps, and normally adding extra steps to a synthesis will reduce your yield because you're gonna every time you add an extra step, you've got to clean up that product, and you're going to lose some product in that process. Remember, that's still way better than just having a random combination of your amino acids where you've got a 25% yield at best. So to summarize what we're doing here, once we know what we want as our N-terminus and what we want as our C-terminus amino acid. So in this example here, we want alanine to be on our N end and our glycine to be on our C end. We have to protect those accordingly. So with our N-terminus, we're going to protect our N group by converting into a carbamate using either Bach 2O or FMOC. And in the case of our C-terminus, we're going to convert that carboxylic acid end into an ester. So in this case here, we're using methanol to make the resulting methyl ester. Okay, so then we can use DCC. Once we've protected those groups, we can use DCC to link them together, as you can see here, and then we can deprotect those groups accordingly. So we can use trifluoroacetic acid to get rid of that Bach group, and we can use a base over here to get rid of that ester. And we're left with our dipeptide in the order that we want. Okay, now that being said, I did point out that, adding these extra steps will affect your yield, and that adds up over time. Like, with a large, when you have a very long peptide, you are still losing, yield with each of those extra steps, and that, adds up. So even if you have a really good percent yield, so let's say we're talking about 95% yield for each reaction, and that's very optimistic, right? With, let's say, doing a tetrapeptide, that combined 5% loss for each reaction yields to a very low percentage. We're talking about like 63% here, with each, because it's not just 4 steps we're talking about, right? We're talking about an additional 2 steps for each amino acid for protecting and deprotecting. So that adds up to give us 9 steps, right? So that's where these losses really add up, and so even with just 4 amino acids linked together, we're dropping down to a 63% yield. Okay, so to get around that, we have another method called a Merrifield synthesis, which cuts down your number of synthetic steps and therefore will give you a better yield overall. Okay, so to do this, you first have to attach your amino acids to a surface of polymer beads. Okay, so we have a polymer strand, or beads that are made of polymer of polystyrene, and they have these groups on them that allow your amino acids, in this case a protected amino acid, to attach via the carboxylate end, okay, via that C terminus end. Okay, so again, note that we do have to protect the our N terminus here to avoid any side reactions between our amino acids. Okay, so we just only have our C terminus available, C termini available, and so the only thing available to react with them are these chlorines. Okay, so here we tack on these our amino acids accordingly. Now we can deprotect those N termini, right? We can get rid of those Bach protecting groups using our trifluoroacetic acid, and then we can bring in a Bach protected amino acid. So the next amino acid in our sequence that's been protected with that Bach protecting group can then be coupled on using DCC. So notice that we've now grown our chain with that Bach protecting group. Okay, now of course we can deprotect that Bach group and and keep on going. Now, obviously you've probably picked up on that we're still protecting and deprotecting those Bach groups on our N terminus of our amino acids, but note that we do not need to protect the C terminus. Okay, the C terminus is completely left open, and because it's kind of constantly being used to extend the chain, and that's what we're reacting with, we don't need to worry about any side reactions. Since we're protecting the nitrogen end, we don't have to worry about our C terminus reacting with a nitrogen that it shouldn't. That cuts down our number of protecting steps, and therefore that increases our yield. Okay, so that basically that increases yield through cutting down the number of steps. The other thing that's really useful with the Merrifield synthesis is purification, right? We've got we're doing this kind of semi heterogeneously, right? We're attaching to these heterogeneous polystyrene beads. But more importantly, when we're done growing our chain, we can just simply cleave off our peptide chain from the beads using hydrofluoric acid. Okay, and you just cut off that peptide chain, and there's your product. You don't have to worry about, again, deprotecting uh, your carbon end or anything like that, or doing anything further. All right. So, for example, so when this was designed, so way back in 1969, ribonucleus, which has 128 residues. All right. So remember the example that we had earlier of four polypeptides, or sorry, of a tetrapeptide, so with four amino acids, and the yield there at best was going to be 63%, and like I said, that's assuming a 95% yield on each individual reaction, which is very optimistic. Here, with something with 128 residues, you're able to get a yield of 17%. So again, that sounds low, but keep in mind, 128 residues, that's a lot of residues. Okay, so that's like really, really good yield on each step. So, this was good enough to get Merrifield the Nobel Prize. Okay, so that is the first level of how proteins form. Okay, so I mentioned that the sequence in which we connect these amino acids is very important, right? And so that is the primary level of protein structure. But there are four levels of protein structure that determine the shape of your protein. Okay, and they're named, as you would expect, primary, secondary, tertiary, and quaternary. So primary structure, of course, as I mentioned, is the sequence of amino acids, the order in which those amino acids are connected together in your peptide chain. The secondary structure has to do with the backbone of that chain. So in other words, the carbon and nitrogen backbone that makes our chain has carbonyl and amine groups on it, and those can engage in hydrogen bonding. Okay, so based on how those are oriented, the backbone of our chain, of our carbon and nitrogen chain, can sort of link up with other similar chains, or even parts of the same chain. It just kind of folds in on itself accordingly. Okay. So you see this in things like alpha helixes or beta pleated sheets. Okay, so you can see here that we can, when these groups start hydrogen bonding with each other, they spiral into a helix, or they sort of weave back and forth into these sheets. Okay. In computer models of proteins, you'll often see these abbreviated by either this curly ribbon for a alpha helix, or using arrows to show the back and forth sheets that are forming. Now, quick note here about different types of beta pleated sheets. You can have the chain weave back and forth like this. That's called anti-parallel, because you have the sequence growing in one direction and then the other relative to each other. Or if you have kind of a loop around, they wind up being parallel to each other. Okay, so that's a parallel beta pleated sheet. All right. So again, yeah, when we look at computer models of these large complex proteins, sometimes having these little abbreviated things to show the secondary structure are very, very useful. Okay. Now, the third level of structure, the tertiary structure, that comes about from the way that your strand folds. Okay, now what determines this is the side groups on your amino acids. They interact with each other, and that's what causes the protein to sort of fold in on itself, or connect to other strands of protein. Okay. So, this, for example, if you have a polar side group, it will interact with other polar side groups. If you have an acidic side group, it will interact with a basic side group, and and so on. Like a nonpolar one will interact with another nonpolar one, and so on. And since this gives a lot, a good chunk of the structure of a protein, messing up with these interactions will cause the protein to get denatured. In other words, to change its shape. which will in turn cause it to change its function. Okay. And by the way, there are lots of ways you can do that. You can do that through heating, right? I kind of mentioned here, like, temperature will denature it, but there's lots of things you can use to interfere with this tertiary level of structure. You can, in the case of those salt bridges, the acid-base interactions, you can mess it up by changing the pH of your solution. You can, you know, do even something as simple as just using mechanical energy. So just like whipping your protein will change its will cause the protein to get denatured. You'll see a lot of these examples oftentimes with cooking, right? So when you fry an egg, that's where the temperature causes your proteins to get denatured, and that you can see that in your eggs albumin changing color, going from colorless to white and opaque, right? In the case of acids, you can see this when you add, lemon juice to milk, right? You can see it kind of like sort of curdling. Or in the case of the agitation, you can see this in forming a meringue, right? If you just whip egg whites, they'll start getting firmer. And again, this is an example of where you can apply mechanical energy to denaturing a protein. Okay, and all of these again work in the same way in that they interfere with this tertiary level of structure. Okay, and then finally, the quaternary level results from multiple chains combining. Again, we kind of use intermolecular interactions between side groups. So again, it's kind of like the tertiary level of structure. It's just on a larger scale. Instead of being sort of intramolecular within a peptide strand, it's intermolecular between multiple strands, multiple sections of protein. Okay, so for example, here we have hemoglobin, and this is made up of 4 different protein subunits. It's the quaternary level of structure that connects those units together. Okay, now based on how these proteins form, how they, their shapes, proteins can have different structures accordingly, or different functions based on their structure. For example, if you get proteins that are a little bit more fibrous because of the way they're arranged, they're known as structural proteins. These include things like hair and nails and, wool and feathers and stuff like that, right? Things that are tissues, so to speak, that are kind of a little bit hardier, like a little tougher. Yeah, basically you have just a lot of strong interactions between these protein chains, and they kind of wrap around each other into long strands or fibers that have some strength to them. Okay. Enzymes have their function also arise from their shape. So enzymes are globular proteins. They're, instead of being strands or fibrous proteins, they tend to exist as clumps. But again, the way they fold in on themselves is important because enzymes are biocatalysts, right? They help carry out reactions. They do that by having a particular substrate fit into pockets in that globular protein. So the shape of that pocket is very important for the reaction that's happening. It allows the protein, the enzyme, to accept the substrate. Basically, you may have heard of a lock and key model. Basically, you need an enzyme that has a pocket that fits the substrate that catalyze, that's the reactant in the reaction it catalyzes. And any other substrates just won't fit in there, and they'll just bounce off. That's kind of an oversimplification. There's really a little bit more nuance to this. The enzyme does have a little give to it, but that's I think a good way to sort of picture it for now. Okay, in a very simplified way. Okay, transport proteins, those are another example. Again, kind of like with enzymes where you've got a pocket, you've got something that allows, whatever you're transporting to get bound to, or relatively weakly bound to your protein so that it can be transported. Okay, so in this example here, with hemoglobin, you've got a heme, you've got this iron ion that's trapped in between here, and it's able to bind to an oxygen. All right, and that's it for this chapter. So let's go through these practice problems. Here's an example with the Henderson-Hasselbalch equation. Again, I don't think you need to stress too much about these. This is kind of a very gen chemy problem, so I'm going to gloss over this one. All right, so again, you're just using the Henderson-Hasselbalch equation and just plugging the numbers in. All right, so let's try to make leucine here. So using the amido-malonate synthesis, you're starting off with your diethyl amino-malonate, and go ahead and try and add on that R group that would give you leucine. Okay, so here's the full mechanism. So here we have our the amido-malonate, right? And of course you treat it with a base that deprotonates that alpha carbon. Now, to make leucine, we want this isopropyl group added on, or sorry, an isobutyl group added on as our side chain. So we're going to use a corresponding alkyl halide here to tack on that alkyl group. Okay, and then of course in the last step we're just going to hydrolyze that, and then we're left with the resulting carboxylate. Actually, I think. The resulting amino acid. I think this is supposed to be a hydrogen here. I believe that's a typo. Okay, so over here we have another essay type question, which again I could probably just gloss over. This is actually a really good example of understanding why the primary level of structure is so important. So if you have a genetic mutation, your genes are basically instructions to your cells on how to synthesize proteins, right? So when you have a mutation, it changes the protein produced on that primary level. It just changes the sequence of proteins, right? Either proteins get switched around, or a particular amino acid is going to be missing, or so on. And just having that one error in the code causes the resulting protein to be completely different, right? Because now, once you've changed that primary level of structure, that's going to go on to affect the the tertiary and quaternary levels of structure, because now your side groups are not in the places that they should be, and so the protein's not going to fold in the way it should, and so on. Okay, so as a result, let's say you've got an enzyme, the pocket that your substrate's supposed to fit in is not the same, and that enzyme doesn't work and doesn't do the job it's supposed to. Okay. So we've looked at proteins. Basically any polypeptide is a polymer, right? You've got a lot of your amino acids are your monomers, and they're linked together to form these polypeptide chains. This is kind of a silly point to make, but the polystyrene beads and the Merrifield synthesis are also polymers, right? They're made of polystyrene. I don't know, that's kind of silly. Again, this is probably not a really important additional practice problem. So you kind of want to, maybe focus on the the homework problems I've suggested on Blackboard as usual. All right, so we obviously have a quiz after this chapter, so if you have any questions and stuff, just let me know, and good luck on your quiz.